All right, and this is going to be, all right, chapter 28, by the way, and we're going to be doing the reproductive system. Now, again, some of what we're going to be talking about today is going to be review. Yay! All right, and we'll talk about some new stuff. All right, for those of you that thought, oh man, I'm going to make it through Bio 210 and 211 and not have to go over the cell cycle and talk about mitosis and meiosis, tricked you. All right, we're going to talk about meiosis here a little bit. Okay, so we're going to first discuss, all right, gametogenesis, and that is going to be the formation of our human sex cells. All right, what's a human sex cell? All right, sperm in men, oocytes or eggs in women. That's simple. Okay, specifically secondary oocytes. Okay, so we talked about this in chapter 4. All right, there's two types of cellular division. There's mitosis and meiosis. Meiosis involves the sex cells, the gametes, all right, and mitosis involves what we call somatic cells. So pretty much every other cell that is capable of cell division is going to undergo mitosis, okay? And our eggs and sperm undergo meiosis, okay? So it's going to be pretty much the same, all right, between the eggs and sperm. There's a few differences, and we'll go through this, all right? So let's quickly, well, I can't say recap because we never talked about meiosis before, all right? But let's quickly just talk about some of the finer points of meiosis, because this could take several classes if I were to go into it in depth. For those of you that have ever, is anyone here taking a biology class in like high school where you talking about? Oh, for some reason it's the first time. Okay, all right, no worries, no worries. All right, so we're going to start off with meiosis. We're going to start off with what we call a diploid parent cell. That's basically a cell that has 46 chromosomes. I'm just going to put CR, okay? All right, so half of those, 23, because 23 plus 23 is 46, all right? Half of those come from mommy, and half come from daddy, okay? So when we're talking about meiosis, we're going to be talking about this diploid parent cell. Right, it's either going to be in the testes or the ovary here, okay? So now, when we undergo meiosis here, we're going to wind up with what we call haploid gametes. What does that mean? That means the total chromosome count in those specific cells will be 23. Okay, don't worry about if it comes from mom or if it comes from dad. We'll discuss that, okay? So a diploid cell is 46. A haploid cell is 23. Are we good so far? All right, I just want to get down some of the basics. So I'm going to be saying some terms and throwing things out there, and I just don't want anyone to be confused about that. Okay, so this next slide shows a quick comparison between mitosis and meiosis, okay? So mitosis, somatic cells, meiosis, gametes, or sex cells. Okay, at the end of mitosis, all right, we talk about this like, for example, in the skin, our keratinocytes, all right? When a keratinocyte divides in the stratum basale, and then one cell stays in that stratum basale layer, and the other one is going to move up into the stratum spinosum. That cell is called the daughter cell, all right? So we have two daughter cells that will be produced, all right, from mitosis. So where we had one cell, all right, now we wind up with two cells that are identical. And when I say identical, genetically identical, same genes, all right, everything, okay? So they're daughter cells, but in this case, the daughter cells are diploid, which means the parent cell had 46 chromosomes. Each daughter cell has 46. Okay? So the daughter cells are diploid. Now this is going to come in later on, all right, crossing over. Think of crossing over as kind of mixing something up. This is how we get genetic diversity. Okay? There's a term that they used to use. I don't even know if they use it anymore. It's called chimerization. Okay? So when we get into meiosis, at the end of meiosis, because there's meiosis 1 and then there's meiosis 2. But at the end of meiosis, the whole thing, we're going to wind up with 4 daughter cells. But they are genetically different from the parent cell. All right? Part of that has to do with this, that crossing over. Okay? But they're different, which is good. All right? But at the end, we'll have 4 daughter cells, and all of those daughter cells will be haploid. What does that mean? They'll have 23 chromosomes, not 46. Okay? 23 is half of 46, so that's what we call it. Haploid. Half. Okay? So when we talk about crossing over, and I'll show you this here in a second, we're going to be exchanging genetic material between chromosomes. All right? But they're going to be genetic material for, like, let's say, hair color or eye color. Like dad might have red hair, like me. And mom could have blonde hair. Okay? And what will happen is the crossing over, all right, the male chromosome will have the red hair, the female chromosome or the maternal chromosome will have blonde hair. What will happen is during the crossing over, they swap. So now the male chromosome will have blonde hair on it, and the maternal chromosome, all right, will have red hair. Okay, I'll show you a picture. Okay, so that's why when we have this crossing over, both parents are going to contribute genes, all right, on one chromosome. It's kind of cool. That's how we get that diversity. That's what we want. Okay, so when we talk about the cell cycle, all right, there's different parts of the cell cycle. Meiosis is one part, okay? Interphase is this part right before cellular division. It occurs primarily before meiosis here. And during this time is when we have to make copies of DNA. Because guess what? We've got to make new cells. We need more DNA. Okay? We can't just split the DNA. All right? So we've got to replicate the DNA. Okay? Does anyone know what that's called? Translation. Okay? We're going to genetically copy the DNA, okay, for each cell. So we now call those replicated chromosomes. All right? So now our chromosomes are made up of 2 what we call sister chromatids. What the heck does that mean? Okay? So here's a chromosome. It looks like this sesame seed stick. Okay? So now what will happen is, all right, during interphase, we're going to replicate that chromosome, and we're going to make another one right here. Whee! And we're going to connect the 2. All right, by this structure, we call it a centromere. So it's going to kind of look like that. Okay? But here's the nice thing. They're going to have identical DNA. All right, so I'll show you a picture here and come right back. So this is what it's going to look like when we're looking at, there we go. So here are our chromosomes, and you can see, I'm going to try not to zoom in too much, okay? So here is the maternal chromosome from mommy and the paternal chromosome from daddy, okay? So what will happen is each one of these chromosomes during interphase will replicate. So now instead of having just one blue thing, we'll have 2 blue things, all right? Instead of having 1 red thing, we'll have 2. Looks like a bow tie almost, okay? So where we had 1, all right, now we made another one, just like it. And it's connected by the centromere here, okay? They're both either maternal or paternal. Okay, so when we go down here, you notice when we get down to these pictures here, we're like, wait a minute. Those aren't all red, or those aren't all blue. I can see on this blue one, there's a little bit of red on there. That's right, because what will happen is we'll have what's called chimerization, and what will happen is that some of these chromosomes will randomly just cross over on one another, and they'll swap genetic information, okay? And then they'll split off, and they'll take the other's genetic information, replace it with their own here. So you can see how this little spot here is blue, and this little one here is red. They switched over, they crossed over. That's how we start swapping genetic information between the maternal and paternal chromosomes there, and that allows us to be more genetically diverse. Okay? So you might have you know, like I keep using hair color. Mom might have blonde hair, dad has brown hair, their kid might have red hair. Okay, because they're swapping. It depends, and I won't get into genetics. Then we're starting to get into dominant versus recessive traits. We won't do that. Just know, all right, that at the beginning we started off with this chromosome. We go in interphase, we replicate it, so we have 2. Okay, because remember we need to make 2 cells. All right, so that's what we're going to see here. We're going to see replication of our chromosomes. Okay, so where we had 1 copy, now we have 2 copies, and we call those 2 copies sister chromatids. They're identical, but just one comes from mom, one comes from dad. Okay, you guys cool with that? Cool. All right, and they're attached by this fella here, the centromere. So now let's jump into meiosis. Okay, so overview of meiosis. We are going to deal with homologous pairs of chromosomes. Right? Homologous pairs are the same exact chromosome. Okay? So we're going to separate these pairs from one another. Okay? So basically at the end of meiosis 1, all right, we're going to separate homologous pairs of chromosomes. That means a pair from mom and a pair from dad. All right? And then we're going to result in 2 cells. And this means that each cell at the end of meiosis 1, all right, is going to have 23 chromosomes of replicated sister chromatids. That's still 46. Because remember, sister chromatids are 2. This is a chromatid here. All right? But we still, those sister chromatids are attached here by that centromere there. Okay? So each of these chromatids have 23 chromosomes on it. Okay? All right. So let's talk about meiosis. There's going to be 4 phases, and I remember it like this: PMAT. So the first phase is prophase 1 because we're in meiosis 1. Okay? So now what we're going to do is we're going to, okay, roll call. Let's find out who's available. We're going to pair up all of the homologous replicated chromosomes that we just replicated, all that DNA that we just copied. We're going to pair it all up now. Okay? And so we're going to form what we call a tetrad. All right, which is 2 sets of 2 sister chromatids. All right, Dr. Kaz, this is getting a little bit crazy here. Let me show you a picture then. This, all right, is what we're talking about. All right, so we've replicated our chromosome here. And we have, all right, so our chromosome is made up of these 2 sticks combined at the center. Okay, so remember, 1 comes from mom, 1 comes from dad. So we just replicated it. So we should have 2. Okay, so we've had crossing over occur already because now we've swapped some genetic information here. So now what we're doing in prophase is we're going to get all of these similar chromosomes and group them together. Hang out with one another. Get them close to each other. Okay? So we're going to get them close to each other. We've already crossed over. And you can see, remember these guys? Remember the centrioles? Centrioles, the only time you really see a centriole is when a cell is ready to divide. That's the only time that we ever really talk about these centrioles. And these centrioles, all right, suddenly appear and they migrate, all right, to opposite ends of the cell because they're going to get ready to form what we call the mitotic, or in this case, it's going to be the meiotic spindle. It's going to shoot out these microtubules here. All right, it's going to attach those microtubules to these chromosomes. Okay, so this is what we're starting to see in prophase 1. Okay, so these homologous pairs are going to start to group up with one another. Okay, they're going to hang out for a little bit. Okay, so we've already seen, all right, in that picture there, but this is where we're going to get that crossing over. We're going to exchange, all right, that genetic material. Same portion of that genetic material on both the maternal and the paternal. Okay, so what that means is I'm just going to swap out hair color, for example, on the male and female, or the maternal and paternal chromosomes, or eye color, or how tall someone's going to be. all right? Or how dark their skin's going to be, or how light their skin's going to be, all right? All that will get exchanged, and it's random. It's not the same thing every time. It just randomly does this. It's part of nature, okay? So, but important thing is, crossing over is going to allow us to have diversity, okay? So now, at this point, we're going to start to break down the nuclear envelope. And you remember the nuclear envelope is to the nucleus what the plasma membrane is to a cell, okay? So the nuclear envelope is that kind of barrier that separates the nucleus from the cytoplasm. So that starts to break down, okay? Now it leads us into metaphase, all right? Which is the next phase here. So now we're going to take those homologous pairs of chromosomes and we're going to stick them at the middle, what we call the cell equator or the midline of the cell. Right? So they just randomly just amble their way over to the midline of the cell. Okay? And some of those chromosomes, and there's no particular order to it, but some of the maternal chromosomes will go to one side. Some of the paternal chromosomes will go to the other. Right? They just randomly do this. It won't be paternal chromosomes on the left, maternal chromosomes on the right. No. It just randomly, one will be on one side of the equator and one will be on the other side. Okay? We refer to this random alignment as independent assortment. Okay? Again, it offers randomness, all right, to the spreading of this genetic information here. Okay? And then our centrioles, all right, are going to start to shoot out these microtubules, all right, from each end, because the centrioles have now migrated to opposite ends of our cell, and they start shooting out these microtubules, and they're going to attach onto the centromere, which is that circular structure at the center of our chromosomes there. So these microtubules will attach onto this thing. They just shoot out onto them. Okay? I'm going to come back to this slide. Let me show you a picture. Whoops. All right, and that's what we're seeing here. So in our metaphase now, all right, here are the centrioles. They've gone to either side. All right, now we've got our chromosomes. They lined up at the center here along the equator. So we've got, all right, paternal chromosome on this side and the maternal chromosome on the other side. But notice we've got another maternal chromosome on the opposite side as this one. So it just randomly sets itself up. But the homologous pairs are always going to be opposite of one another. Okay, always opposite. So these are relatively the same. They exchange the same genetic information, all right, for hair color. Okay. So now we're going to get these microtubules that are going to shoot out, and what we're going to see is we're going to start to pull the cell apart at the center here. Okay, and you can see here when we get into anaphase, all right, these microtubules start to pull the chromosomes back away from each other. Where am I? Interphase. Okay? So now we start to separate our homologous pairs, and they're going to start to go towards the opposite ends of the cell. All right? That's hence what we call reduction division here, because what we've done now is we have reduced the number of chromosomes by half. Now, we're not going from 46 to 23. That doesn't happen yet. But what we've done is, here's the cellular equator here, okay? And on one side of the equator, we have a chromosome. And on the other side, we have the same exact chromosome. But remember, one comes from dad, one comes from mom. But both of them each have 46 chromosomes. So if you add that together, what is that? 92. Okay? So you had 92, but now that when we separate the cell, all right, now we have just 46 in each cell. I don't want you to get stuck on the value 92. I'm just throwing math out there. So you understand when we say how we have half the number of chromosomes, I don't want you to think it's haploid. It's not. All right, haploid is 23 chromosomes. Diploid is 46. Okay, just don't get confused. So that means each chromosome still has the 2 sister chromatids. That's this. That's one chromatid. Here's the other chromatid. The centromere at the center. Remember, each chromatid is how many chromosomes? 23. 23, 23, which is total 46. Okay? All right, so that's what we're seeing here in the anaphase. We're starting to pull those back. All right, so you've got 46 on this side, 46 on this side. All right, and then finally, telophase, all right, is when we actually get, all right, the cells are almost fully pulled apart from one another. And so now we're going to package up, all right, our chromosomes back into their nuclear envelopes. Okay? And so the daughter cells now are going to have 23 original chromosomes, but also 23 replicated chromosomes. Okay? So we'll still have the 2 sister chromatids because they're bound together still. Because guess what? We got to do this all over again. But it's here in meiosis 2 that we're going to separate the sister chromatids from one another. So in meiosis 2, what we're going to do is we're going to take this structure here and we're going to split it. We're going to chop it right down the middle. We're going to get rid of that centromere. So that means we'll have 2 separate chromatids in each cell. So you'll have one over in this cell, one over here. Which I will talk about in another class at another time. Okay? All right, let's take a break here. And what I might do is talk about this a little bit in lab, and then we'll do a little bit of a mock quiz.